The disease we see in a patient is expressed through cells, and beneath every cell lies the same starting point: the genome. The human genome contains about 3.2 billion base pairs. That is an enormous amount of DNA. But here is the first surprise. Only about 1.5% of that genome directly codes for protein. That protein coding fraction contains roughly 20,000 genes. 20,000 may sound like a large number, but then consider a worm. A worm has fewer than 1,000 cells, and yet it carries about the same number of protein coding genes as a human being. So gene number alone cannot explain human complexity.
Having more complex tissues does not simply mean owning more genes. The deeper difference lies in how those genes are used. Now we come to the other 98.5% of the genome. For years, much of it was loosely described as junk, but that description is misleading. A large proportion of the genome is transcribed at some point, even though it does not become protein. And much of this non-coding genome performs regulatory work. It contains regions that help determine when a gene is expressed, where it is expressed, how strongly it is expressed, and how long that expression continues. This brings us to the central idea of the entire video. Nearly every nucleated cell in the body carries the same complete genome. A liver cell and a neuron own essentially the same DNA sequence. The neuron does not possess a special set of neuronal genes that the liver cell lacks, and the liver cell does not own a separate liver genome. They possess the same genetic library. What makes them different is which parts of that library they use. A neuron keeps neuronal programs active and most liver programs silent. A liver cell does the opposite. So cell identity does not come mainly from gene content. It comes from gene regulation. The difference between one cell type and another is largely a difference in which genes are open, which are closed, which are transcribed, and how much protein each gene ultimately produces. And this matters enormously in pathology. A disease does not always require the coding part of a gene to be destroyed. The protein sequence may remain completely normal. The problem may be that the gene is expressed at the wrong time, in the wrong tissue, at the wrong level, or not expressed at all. Many disease-associated genetic variants are found outside protein coding regions. They alter regulatory elements rather than the structure of the protein itself. And cancer, at its root, is often a disease of regulation. Growth genes remain active when they should switch off. Tumor suppressor genes become silent when they should remain available. Developmental programs are reactivated in the wrong context. The cell does not necessarily lose the correct books. It begins reading the wrong ones. A useful way to picture the genome is as a vast library. Every cell owns the full collection. A cell is not defined by possessing different books. It is defined by which books are open on the desk, which pages are being copied, and which volumes remain locked away. Much of disease is not a missing book. It is the wrong book left open. The one sentence to keep. The human genome contains about 3.2 billion base pairs, but only about 1.5%, roughly 20,000 genes, codes for protein, while much of the remaining genome regulates when, where, and how strongly genes are expressed, meaning that cell identity and much of disease arise from regulation rather than gene number. Now that raises the next question. If so much of the genome is transcribed but never becomes protein, what is all that RNA actually doing? We usually learn RNA as a messenger. DNA is transcribed into messenger RNA, and messenger RNA is translated into protein. But not every RNA is a messenger. A large class of RNA molecules never becomes protein at all. Instead, the RNA itself performs regulatory work. The best-known example is the microRNA. A mature microRNA is tiny, only about 22 nucleotides long. But its regulatory reach can be enormous. To understand what it does, first follow how it is made. The process begins in the nucleus. A longer precursor RNA is transcribed. An enzyme called DROSHA begins processing it inside the nucleus. The partially processed RNA then moves into the cytoplasm. There, a second enzyme called dicer cuts it again. This produces the short, mature microRNA. But the microRNA does not act alone. It is loaded into a protein complex called RISC. RISC is the silencing machinery. The microRNA gives that machinery its address. Its nucleotide sequence allows it to recognize a complementary sequence in a target messenger RNA. RISC then carries the microRNA to that matching message. Once the target is recognized, the messenger RNA is either blocked from being translated or is degraded. The gene has already been transcribed. The messenger already exists. But the message is prevented from producing its normal amount of protein. This is post-transcriptional regulation. The control happens after transcription, but before the final protein output is completed. And this system is not a minor side pathway. One microRNA can regulate hundreds of different messenger RNAs, and around 60% of human protein-coding genes are estimated to be under some degree of microRNA control. That means one small RNA can influence an entire cellular program. It can adjust metabolism, growth, differentiation, survival, and the response to stress. MicroRNAs often do not behave like simple on and off switches. They usually tune protein production. They lower one message strongly, another moderately, and another only slightly. This allows the cell to coordinate many targets at once without completely silencing all of them. MicroRNA regulation is therefore more like a set of dimmer controls than a row of light switches. But microRNAs are only one part of the non-coding RNA world. There are also long non-coding RNAs. By definition, these are longer than 200 nucleotides, and they can work in several different ways. Some act as scaffolds. They bring several proteins together into one functional complex. Some act as decoys. They bind a regulatory molecule and prevent it from reaching its normal target. And some act as guides. They carry regulatory proteins to a specific region of the genome. This allows long non-coding RNAs to influence entire chromatin regions, not merely individual messenger RNAs. The classic example is XIST. XIST is a long non-coding RNA involved in X chromosome inactivation. It spreads across one X chromosome in female cells. It helps recruit silencing machinery, and that chromosome becomes largely transcriptionally inactive. So non-coding RNA can operate at different scales. A microRNA can tune individual messenger RNAs. A long non-coding RNA can help organize or silence an entire chromosomal region. Now consider what happens in cancer. A microRNA has no fixed identity as good or bad. Its effect depends entirely on what it targets. If a microRNA silences a tumor suppressor, the cell loses a brake on growth or survival. That microRNA is acting like an oncogene. An oncogenic microRNA is often called an OncomiR. But if a microRNA silences an oncogene, it reduces a growth-promoting signal. In that setting, the microRNA behaves like a tumor suppressor. The same class of molecule can therefore promote cancer or oppose it. The difference lies in the target. Why regulate genes through RNA when transcription factors already exist?
Because RNA-based regulation offers several advantages. It can be rapid. It can be reversible. It can be finely dosed. And one microRNA can coordinate many related targets at the same time. Transcription factors help determine which genes are transcribed. MicroRNAs help determine how much protein finally emerges from those transcripts. If transcription factors are the switches that activate genetic programs, microRNAs are the dimmer dials that tune their final intensity. A cancer cell is not always missing a switch. Sometimes it has simply turned the wrong dials. The one sentence to keep.
Non-coding RNAs regulate genes directly as RNA. MicroRNAs are approximately 22 nucleotide guides processed by Drosha and Dicer and loaded into RISC to silence matching messenger RNAs, while long non-coding RNAs act as scaffolds, decoys, or guides that can regulate entire chromatin regions, allowing both classes to function as oncogenic or tumor-suppressive regulators. Some non-coding RNAs tune individual messages. Others, like XIST, help silence entire regions of the genome. But what does it physically mean for a region of DNA to be open, closed, active, or silent? To answer that, we have to look at how the genome is packaged. The cell begins with a physical problem. If the DNA from one human cell were stretched out, it would measure approximately 2 meters, and all of it must fit inside one microscopic nucleus. So the DNA cannot remain as a loose, extended thread. It must be packaged. The DNA wraps around proteins called histones. A segment of DNA wrapped around a core of histone proteins forms a nucleosome. Nucleosomes then organize into progressively higher levels of packing. Together, the DNA and its associated proteins form chromatin. But chromatin is not merely a storage solution. The way the DNA is packed carries biological information. Packing determines access. If a region is loosely packed, the transcription machinery can reach it more easily. If it is tightly condensed, the genes inside that region become difficult to access. Open, relatively loose chromatin is called euchromatin. Euchromatin is generally accessible and transcriptionally active. Condensed chromatin is called heterochromatin. Heterochromatin is generally inaccessible and transcriptionally silent. This gives us the physical explanation for a question we raised in the first board. How can a neuron and a liver cell contain the same genome but behave completely differently? They keep different parts of that genome open. The neuron maintains access to neuronal genes. The liver cell maintains access to hepatic genes. Each cell closes many of the programs that belong to other lineages. The sequence is largely the same. The accessibility is different. And that difference in accessibility is controlled by a layer of regulation called epigenetics. Epigenetics refers to heritable changes in gene expression that occur without changing the underlying DNA sequence.
The text remains the same. What changes is how the text is packaged and read. These changes can be maintained when a cell divides. A daughter cell can inherit not only the DNA sequence, but also much of the pattern of which regions should remain active or silent. This helps preserve cell identity. A liver cell divides and produces another liver cell. It does not have to rediscover its identity from the beginning after every division.
Its pattern of gene usage is carried forward. And yet, epigenetic regulation is not completely permanent. It remains potentially reversible. That balance is what makes epigenetics so useful. The cell needs the DNA sequence to remain stable, but it needs the use of that sequence to remain flexible. During development, cells must activate one program and silence another. When a signal arrives, a cell may need to open a set of genes temporarily. When the signal disappears, those genes may need to close again. The cell must change expression without repeatedly rewriting its genome. Epigenetic regulation provides that rewritable layer over a fixed sequence. Several types of chemical marks help control chromatin. One important category is histone modification.
Histone acetylation is strongly associated with open, active chromatin. Adding acetyl groups weakens the interaction between the negatively charged DNA and the histone proteins around which it is wrapped. The chromatin becomes less compact, and transcription factors and other components of the transcription machinery gain easier access. Removing those acetyl groups generally favors a more closed chromatin state. Histones can also carry methyl groups and other modifications. The effect of histone methylation depends on which amino acid is modified and how many methyl groups are added. Some histone methylation marks are associated with activation. Others are associated with repression. So the important principle is not simply that methylation always means silence. The location and context of the mark determine its meaning.
A second major mechanism is DNA methylation. Here, methyl groups are added directly to cytosine bases. This occurs especially at CpG sites, where a cytosine is followed by a guanine. When CpG-rich promoter regions become heavily methylated, transcription is usually suppressed. The methylated DNA recruits proteins that help maintain a condensed, silent chromatin state. The gene may still be physically present. Its coding sequence may be completely normal, but it becomes functionally unavailable. This distinction is essential in pathology. A gene can be genetically intact and functionally absent. Cancer cells exploit this. A tumor suppressor does not always have to be deleted or mutated. Its promoter may become abnormally methylated. The gene is silenced.
The protein is no longer produced in the amount required, and the cell loses an important break, even though the DNA sequence of the gene itself remains intact. This is one reason epigenetic change is so important clinically. A mutation alters the text itself. Reversing that change is difficult. But epigenetic silencing alters how the text is read. And because the sequence remains present, the possibility exists that the gene may be reactivated. This is the basis of therapies designed to inhibit abnormal DNA methylation or alter histone modification. Epigenetic reversibility turns a mechanism of disease into a potential therapeutic opportunity. A useful way to picture the relationship is to return to the library.
The DNA sequence is the printed text. Chromatin determines whether the book is open on the desk, closed on the shelf, or locked away in storage. Epigenetic marks, including histone modifications and CpG DNA methylation,
can help decide where the book is kept. Cancer can silence a tumor suppressor without tearing out the page. It can simply lock the entire volume in a cabinet. The one sentence to keep. DNA is packaged around histones into nucleosomes and chromatin, where open euchromatin is generally accessible and active, while condensed heterochromatin is generally silent, and epigenetic marks, including histone modifications and CpG DNA methylation, create heritable but reversible changes in expression without altering the underlying sequence, allowing cancers to silence intact tumor suppressor genes.
Epigenetics can change how the genome is read without changing a single base of DNA. But what if the sequence itself is defective? Can we enter the genome, locate one exact region, and deliberately change the text? Until recently, studying the genome mostly meant reading it. We could identify a mutation. We could connect that mutation to a disease. We could observe that patients carrying the mutation developed a particular phenotype. But actually entering a living cell, locating one exact DNA sequence, and changing it was a completely different challenge. CRISPR changed that. CRISPR-Cas9 is a programmable system for cutting DNA at a chosen location. And humans did not invent the system from nothing. We borrowed it from bacteria. Bacteria are constantly attacked by viruses. To defend themselves, they evolved systems capable of recognizing foreign viral DNA and cutting it. Scientists realized that this molecular defense machinery could be redirected.
Instead of sending it after a viral sequence, we could program it to recognize a DNA sequence chosen by us. The system depends on two main components. The first is a guide RNA. The guide RNA contains a short sequence designed to match the selected target in the genome. It gives the system its address. The second component is the Cas9 enzyme. Cas9 is the molecular cutting tool. The guide RNA directs Cas9 through the genome. When the system finds the matching DNA sequence, Cas9 cuts both strands. It creates a double-strand break. And this is where an important distinction must be made. Cas9 makes the cut. But Cas9 does not perform the entire edit by itself. The cell recognizes a double-strand break as a dangerous lesion, so the cell immediately activates its own DNA repair machinery. What happens during that repair determines the final outcome. Sometimes the cell simply rejoins the broken ends. That repair may introduce small errors. A few nucleotides may be inserted or deleted. The reading frame may be disturbed. The resulting gene may no longer produce a functional protein. In this way, CRISPR can be used to disable or knock out a gene. In other situations, a carefully designed DNA template can be supplied. The cell may use that template while repairing the break. This creates the possibility of rewriting the sequence more precisely. A harmful mutation may be corrected. A missing sequence may be inserted. Or a specific experimental alteration may be introduced. So CRISPR is not merely a pair of molecular scissors. It is a targeting system combined with the cell's own repair machinery. The guide RNA determines where the system goes. Cas9 creates the break. And the repair process determines whether the gene is disrupted or rewritten. Why does a gene editing tool belong in a pathology course? There are two major reasons. The first is that CRISPR transforms how we study disease. Suppose a particular mutation is repeatedly found in a tumor. That association alone does not prove that the mutation caused the malignant behavior. Using CRISPR, scientists can introduce the mutation into a controlled model and observe what changes. Does the cell begin dividing faster? Does it resist apoptosis? Does it invade? Or the mutation can be removed from diseased cells. If the abnormal phenotype disappears, the causal relationship becomes much stronger. CRISPR allows pathology to move from observation to experimentation. It lets us test whether a genetic alteration truly produces a disease mechanism. CRISPR also allows us to create disease models. Cells can be engineered to carry the same mutation found in a patient. Organoids can be built from edited cells. Animals can be given specific genetic alterations. Researchers can then follow the disease from the first molecular change to the final cellular and tissue morphology. These models are now central to experimental pathology. The same technology also creates the possibility of gene therapy. If a disease is caused by a harmful genetic sequence, it may be possible to disable that sequence. If a functional sequence is missing, it may be possible to restore it. And if a mutation can be corrected, the disease may be treated at its genetic origin rather than only at the level of its consequences. That is the promise. But the second reason CRISPR belongs here is even more fundamental. The double-strand break that CRISPR creates deliberately is the same basic lesion that pathology produces accidentally. Ionizing radiation can cause double-strand DNA breaks. Reactive oxygen species can damage the genome. Replication failure can leave chromosomes broken. Defective repair machinery can allow those lesions to persist. In CRISPR, the break is targeted and controlled. In disease, it is uncontrolled. But the cell recognizes the same emergency. It activates DNA repair pathways. It may arrest the cell cycle. And if the lesion cannot be repaired, p53 may direct the cell toward apoptosis. So CRISPR provides a controlled way to study one of the most dangerous forms of genomic injury. It also reminds us that gene editing is not perfect. The first major challenge is accuracy. A guide RNA is designed to recognize one target.
But similar sequences may exist elsewhere in the genome. If Cas9 cuts one of those unintended sites, that is an off-target edit. An attempt to correct one lesion could therefore create another. The second major challenge is delivery. Editing cells in a laboratory dish is relatively straightforward. Delivering the guide RNA and Cas9 into the correct cells inside a patient is far more difficult. The system must reach the target tissue. It must enter enough of the relevant cells. It must avoid unnecessary exposure of other tissues. And it must do all of this safely. Even when the correct cells are reached and the correct sequence is cut, the cell's repair response may not always produce the intended result. The editing system can choose the location, but the biology of the living cell still helps decide the outcome. A useful way to picture CRISPR is as a molecular search and edit system. The guide RNA is the search instruction. Cas9 is the blade that opens the text at the selected line. And the cell's own repair machinery performs the rewriting that follows. But unlike a computer document, the genome is alive. The editor can choose where to intervene. It cannot completely command how the cell responds. The one sentence to keep. CRISPR-Cas9 uses a programmable guide RNA to direct the Cas9 enzyme to a chosen DNA sequence, where it creates a double-strand break that the cell's own repair machinery can use to disable or rewrite the gene, making CRISPR a powerful tool for disease modeling, gene therapy, and the controlled study of pathological DNA damage.
We began with the genome as a library shared by almost every cell. We saw that human complexity does not come simply from owning more genes. It comes from controlling how those genes are used. Non-coding RNAs tune the messages produced from the genome. Chromatin and epigenetic marks determine which regions can be reached and read. And CRISPR now allows us to target and change the sequence itself. The genome is the information system of the cell. But information alone cannot remain alive. The cell must preserve an inside that is different from the world outside. It must control what enters, what leaves, which ions remain separated, and which signals are allowed to cross. For that, the cell needs a border.
That border is the plasma membrane.